A typical fungus consists of a mass of branched tubular filaments enclosed by a rigid cell wall. The filaments are called hyphae. They branch repeatedly into a complicated radially expanding network called the mycelium, which makes up the thallus or the undifferentiated body of a typical fungus. The mycelium grows by utilizing nutrients from the environment, and upon reaching a certain stage of maturity forms either directly or in special fruiting bodies the reproductive cells called the spores. The spores are released and dispersed by a wide variety of passive or active mechanisms. Upon reaching the suitable substrate, the spores germinate and develop hyphae that grow, branch repeatedly, and become the mycelium of the new individual. Fungal growth is mainly confined to the tips of the hyphae, and all fungal structures are therefore made up of hyphae or the portions of hyphae. In this video, aspects related to the thallus organization in fungi is discussed in detail. The body of the fungus is called as the thallus. There are two types of thalli in fungi. One is Eucarpic thallus and another one is Holocarpic thallus. In Eucarpic thallus, the thallus is differentiated into the vegetative part which absorbs nutrients and reproductive part which forms the reproductive structures. In Holocarpic thallus, the thallus does not show any such differentiation into vegetative and reproductive structures. After a phase of vegetative growth, it gets converted into one or more reproductive structures. Hyphae is a tubular transparent filament usually branched, composed of outer cell wall and the cavity filled with the cytoplasm. The hyphae are divided into many compartments or the cells by the formation of cross walls called septa. If the septa is absent, then the hyphae is called as Coenocytic or aseptate. Septa, it occurs in the thallus of all filamentous fungi to cut off the reproductive cells from the rest of the hyphae, to separate off the damaged parts, or to divide the hyphae into regular or irregular compartments or the cells. The hyphal mass or the network of hyphae constitute the body of the fungus, that is the thallus, and is called as the mycelium. In case of monokaryotic mycelium, it is uninucleate. The mycelium contains single nucleus that usually forms a part of haplophase in the life cycle of the fungus. In case of dikaryotic mycelium, that is binucleate mycelium, the mycelium contains a pair of nuclei, that is the dikaryon, which denotes the diplophase in the life cycle of the fungus. The heterokaryotic mycelium contains nuclei of different genetic constituents. The fungal cell contains more than 2 nuclei. In case of the homokaryotic mycelium, the mycelium contains genetically identical nuclei. Specialized somatic structures. First one is the rhizoid. A rhizoid is a short root-like filamentous outgrowth of the thallus, generally formed in tufts at the base of small unicellular thalli. It serves as the anchoring or attachment organ to the substratum and as an organ of absorption of nutrients from the substratum. They are short, delicate filaments that contain protoplasm but no nuclei. Rhizoids are common in lower fungi like Cartidomycetes, Oomycetes and Zygomycetes. In some species, the rhizoids may be branched and is called as rhizomycelium. On rhizomycelium, numerous sporangia develop. Such thalli are called as polycentric. That is, they form several reproductive centers instead of a single one where the thallus is termed as monocentric. Aprosorium. The aprosorium is a simple or lobed structure of hyphal or germ tube and pressing organ from which a minute infection peg usually grow and enter the epidermal cell of the host. It helps the germ tube or hypha to attach to the surface of the host substrate. These apresoria are formed from germ tubes of uridinales, that is the rust fungi, erysipelas, that is powdery mildew of fungi, and other fungi in their parasitic or saprophytic stages. Hastoria. Hastoria are special hyphal structures or outgrowths of somatic hyphae sent into the cell to absorb nutrients. The hyphal branch, said to function as hastoриum, becomes extremely thin and pointed. While piercing the host cell wall, it expands in the cell cavity to form a wider, simple and branched hastoриum. The hastoria may be knob-like or balloon-like in shape, elongated or branched like a miniature root system. Then hyphopodia. It is a small appendage with one or two cells in length on the external hyphae and function as absorbing structures. The terminal cell of the hyphopodium is expanded and rounded or pointed. The aggregations of hyphae and tissues. First one is the mycelial strand. The mycelial strands are the aggregates of parallel or interwoven undifferentiated hyphae which adhere closely and frequently anastomosed or cemented together. Mycelial strand formation is quite common in Basidiomycetes and Ascomycetes as well as in Deuteromycetes. The mycelial strand form the familiar spawn of the cultivated mushrooms. The mycelial strands are capable of translocating materials in both the directions. They are believed to afford means by which a fungus can extend and establish food base and colonize in a new substratum by increasing the inoculum potential of the fungus at the point of colonization. Rhizomorphs. It is an aggregation of highly differentiated hyphae which often darkly pigmented. They grow faster than the mycelial strands. The growing tip of the rhizomorph resembles that of a root tip. The fungus may spread underground from one root system to another by means of the rhizomorphs. These root-like aggregations are found in the honey fungus. Then fungal tissues. During certain stages of the life cycle of the most fungi, the mycelium becomes organized into loosely or compactly woven tissue called plectenchyma. There are two types of plectenchyma, namely prosenchyma and pseudoparenchyma. The prosenchyma, when the tissue is loosely woven and the hyphae lie parallel to one another, it is called as prosenchyma. In case of the pseudoparenchyma, it consists of closely packed, more or less isodiametric or oval cells resembling the parenchyma cells of the vascular plants. In this type of tissues, the hyphae lose their individuality and are not distinguishable. Then stroma, that means a mattress. A stroma is a compact somatic structure or hyphal aggregation similar to a mattress or a cushion on which the fortifications of the fungi are usually found. Sclerotium. The sclerotium is a resting body formed by the aggregation of somatic hyphae into dense, rounded, flattened, elongated or horn-shaped dark masses. The sclerotia on germination may produce directly the mycelium. A mycorrhiza is a symbiotic association between a fungus and a plant. The term mycorrhiza refers to the role of the fungus in the plant's rhizosphere zone. The mycorrhizae play an important role in plant nutrition, soil biology, and soil chemistry. In mycorrhizal associations, the fungus colonizes the host plant's root tissues either intercellularly or extracellularly. The association is normally mutualistic. The mycorrhiza are commonly divided into ectomycorrhiza and endomycorrhiza. In ectomycorrhiza, it consists of hyphal sheath or a mantle covering the root tip and the hortic net of the hyphae surrounding the plant cells with the root cortex. In some cases, the hyphae may also penetrate the plant cells, in which case the mycorrhiza is called as ectendomycorrhizae, that is outside the root. Endomycorrhizae, also called as arbuscular mycorrhizae, have a hyphae that penetrate the plant cells, producing dichotomously branching invaginations, that is the arbuscules, as a means of nutrient exchange, which are often balloon-like storage structures termed as vesicles.